TWiM listeners, do you have science you want to share? Microworld.org is the best place to post your microbiology-related news articles, pictures, videos, papers, and more. Sign up for a free account at microworld.org/join and start reaching thousands of microbiology enthusiasts just like yourself.
This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 82, recorded on July 3rd, 2014. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Howdy. How you doing?
I am well. Are you still in a drought state over there?
We are in a drought state, and I'm afraid this is serious, and I don't know the answer.
I wish yesterday we had a torrential downpour. The roads were flooded up to 2 feet of water.
Really?
Wow.
I wish I could have sent you some of that.
That would have been great.
The lightning was striking on either side of my car on the way home.
Thanks.
It took me 2 hours to get home, which should normally have taken an hour. Yeah, it was flooding everywhere. It was a mess. It was a really spectacular storm. Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello, everyone.
How's your summer going?
Great. Too fast.
It's going to be over soon.
I'm in shock that it's July.
Yeah. When you hit June, that's it. It's going to go really fast. Before you know it, the undergrads will be back.
And I'll be talking football.
Oh my, yes. Football starts in August, right?
It does.
Wow. Maybe this season you guys will be really good.
Let's talk about something else.
Okay. Have you been playing golf this summer?
Of course.
You have golf courses in Ann Arbor, I guess, right?
I do. I just, I had a personal best recently. I shot a 44 on my course.
Nice.
So for 9 holes, of course.
I was going to say 9 holes, yeah.
Yeah, and I finished with a birdie. So that doesn't happen very often, but it felt great.
Hey, did you ever think about the golf course microbiome when you're out there playing? I guess not. Okay.
I don't.
Also joining us today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
How's Charleston?
We're dodging a hurricane, thankfully. Hurricane Arthur is off our coast, and we're occasionally getting some minor squalls come in. It's about 100 miles, and it looks like it's going to hit the North Carolina coast sometime.
Isn't it kind of early for hurricanes?
It's early, and the folks in New Jersey are blessed in that they think the hurricane's going to run out of energy when it hits the cold water. So, of course, everyone is very much concerned after Sandy. And it's good that it's early in the season because the coast is cold still. It hasn't gotten all the summer heat, and so that'll poop out the energy. And so the storm will hopefully be pretty benign.
Because I'm going away. I'm going to Australia on Friday, which is tomorrow.
Ooh.
Great.
My family will be down the Jersey Shore on their own. So I hope they don't get a hurricane because they would need me to rescue them.
Of course they would.
I would come to the rescue.
Well, just give your son strict orders to batten down the hatches.
All right. Today we have 2 papers for your educational pleasure, I should say. I was on the elevator the other day with 2 people whose names all of you should know, Richard Axel and Tom Maniatis. You guys all know those names?
Of course.
Oh, yes.
Okay. So they were both here at Columbia, and they were talking. One of them was saying, I really like when people explore new things like David Baltimore giving people antibodies, blah, blah, blah. And he looked at me and said, what are you preventing, Vinny? And I said, I'm preventing boredom.
Ignorance.
Ignorance. You're preventing ignorance.
That too. That was a good answer, right?
That was a good answer.
You bet.
So that's what we do here on TWiM. We provide information in a easily digestible way. And speaking of digestion, Elio is going to start us off with our first paper.
All righty. Let me tell you the title of the paper is Grow EL from Endosymbiont Buchnera aphidicola betrays the aphid by triggering plant defense. And it's by Chowdhury, Atamian, Shen, Briggs, and Kaloshin, who are from the University of California, Riverside, and the University of California, San Diego. And this is a paper about plant defenses and how they are overcome, well, not exactly, how they're set off by the presence of parasites, in this case aphids, with a twist. And the twist is that aphids carry many aphids, most aphids, I think, carry an obligate endosymbiont, a bacterium called Buchnera aphidicola. And they carry it for a good reason, because aphids are sap feeders. They feed on sap of plants, and saps are mainly sugar solutions. They are thin on amino acids and other things. And if that's all they ate, and they can't make all that amino acids, they would starve. So instead, they have acquired, way back, they acquired an endosymbiont, a bacterium that can make those amino acids and supply the host with them. So, and in fact, it's quite a bit of nice arithmetic, because they supply about 10 amino acids which are required by the aphids, and the aphids turns around and makes the other 10 amino acids and supplies them to the bacterium. So these guys can count to 10. Which is pretty neat. Okay. Anyhow, so that's the background. The background is that a protein called GROW-EL, which we'll talk about, is a chaperonin, which we'll talk about, from the endosymbiont triggers host defenses. In other words, it betrays, as the title says, betrays the aphid by telling the plant that here it is, and this is a signal for trouble, for the trouble for the aphid, and therefore for the bacterium.
Can you imagine that after providing the aphid with 10 amino acids, then the bacteria betray them?
I know. That's what the title is.
Exquisite.
But, on the other hand, bacteria don't have quite that kind of volition.
Yes.
So I think calling it betrayal is a bit of anthropocentrism.
Absolutely.
But we like anthropocentrism because they're fun, right? So we'll live with that. But before I do that, before I go into this paper, which is a very nice paper, came out in PNAS, and it's quite readable and straightforward. Before that, I'd like to say something about plant defenses, and there's a reason for it. Namely, that until recently, I knew very little about. I still don't know much about it, but I know a little bit more than I did until a few days ago. And what's surprising, and it wouldn't be surprising to anybody in the know, but it may surprise some of our listeners, is the amount of detailed, wonderful information that has become available about how plants defend themselves against invaders. This is a sophisticated subject which has really quite some, let's call it mechanistic penetrance. It enters into biochemical, molecular mechanisms in quite some detail. This is in good part because the model organism in plants has become Arabidopsis thaliana, which is a cress, relative of the mustard plants. And it is so easy to manipulate from the point of view of genetics and molecular studies that it's become the model system. So a lot is known about in the infections of Arabidopsis. And as I say, it serves as a model system. But let me generalize a little bit first. Is that okay?
Mm-hmm.
All right. So plants, they succumb to infections. In fact, we know that from history, infections that have caused enormous hardship on humans include the Irish potato famine, which is caused by a fungus-like organism, the oomycete, Phytophthora infestans. A whole banana cultivar called Gros Michael was lost in the 1920s to a disease called Panama disease caused by a fungus called Fusarium. And there are current epidemics spreading through Africa and other parts of rust fungi that affect wheat and other plants. In general, chestnut blight, an example of how we almost eradicated a major tree species in North America. We didn't, but it happened. Anyhow, turns out that plant infections reduce crops by a very, very high, significant extent. It amounts to about 10% in the developed countries and 20% in the less developed countries. That is a huge amount of agricultural loss due to infections. So the subject is worth looking into it. And it turns out that plants have important and significant defense mechanisms. One doesn't think of plants in those terms because certainly plants don't have phagocytic cells, they don't make antibodies, they don't have lymphatic systems and spleens and things like that help them develop an immunity like we know it, be it innate or acquired. However, they have a lot of defenses, and some are we can call innate immunity, have to do with the structure. They're tough. It's hard to penetrate a plant cell. Plant cell walls are surrounded by, are made of very tough polymers, and so that certainly is a defense. Also, plants contain antimicrobial chemicals like glycosides, saponins, antimicrobial peptides, in other words, defensins. They have enzyme inhibitors and so forth. So these are preformed structure, defense mechanisms, but they have also inducible ones. And one of them is what happens when you hurt a plant, a little tree, for instance. It'll make a callus, it'll make a scab, a scar, if you wish. That's made by polymers called callose, and of course suberin, lignin, of course, and they grow out a piece of the plant that looks different and is in a way to circumscribe the lesion. Also, antimicrobial chemicals are made as in response, that is, in an inducible way. They include reactive oxygen species like hydrogen peroxide and complex compounds which have funny names like phytoalexins. So there is really a response, an inducible response by plants, and some become very interesting. In fact, in one mechanism that plants have to defend themselves is to release chemicals that attract beneficial bacteria to fight off infections. We got to talk about that sometime. It's called endophyte resistance. Not for today. Let me go on. Most of what's known about this is in fact from Arabidopsis and a bacterium called Pseudomonas syringae. So please be cautious in thinking about this story, because it is really limited to almost, not entirely, but almost entirely to a single model system. And of course, there are many plants in the world, and so this may not be a good way to think of it. So let me give a little talk, a little primer on plant immunity. First of all, the immune system in plants recognizes the presence of invaders in a way which is remarkably similar to that of animals. It works via recognizing what are known as pathogen-associated molecular patterns, or better, microbial-associated molecular patterns, PAMPs or MAMPs. These are things on the surface of, say, bacteria and viruses. They include things like the protein flagellin, which is what bacteria flagella are made of, certain form of viral RNAs and so forth. All of these are recognized on the surface of animal cells by receptors. And these are called pattern recognition receptors, or PRRs. They're on the surface of cells. And the same thing is true on plant cells. So in fact, that our plant cells have a receptor to recognize bacterial flagellin, very similar to that in animal receptors. And it is similar in that the protein is, these are proteins, the receptors are proteins, which are leucine-rich repeat kinases. So there is a motif, leucine-rich repeats, which is found in certain kinases, and this is found both in the receptors for the PAMPs in animal cells and in plant cells. Isn't that neat?
It's cool. Yep.
All right. So plants know that there are pathogens around, and they do that. Then the response that is set off by the plant is, in this case, is called PTI, which stands for PAMPs triggered immunity. So just like in animals, plants can set off this kind of immunity, and they set off they make a whole bunch of chemicals that are inimical to the life of the invader. However, the pathogens know that this is happening, and they make a second stage of response, namely, they make effectors to inhibit the PTI, to inhibit this PAMPs triggered immunity. And so they say, you are fighting me, I'm fighting you back. And this second layer of defense is called effector. But then the plants respond to that. So there's a second layer of defense mechanisms, which is you send me an effector that tells me that you don't like what I'm doing, I'm going to fight you back.
Sounds like ping pong. Sounds like
exactly.
Or an arms race.
An arms race, whichever. And this is very typical of what happens in animals, where we know this to be the case, and it's true for plants. So these are general, I would say, not only there's a general similarity between plant immunity and animal immunity, but it goes down to fairly molecular detail. It's very similar in both cases. So we need to know about this, and this is really, worth knowing. So this goes back and forth. There is, to summarize what I just said, plants have receptors that recognize the presence of invaders. They set off an immunity to that. This is the PTI immunity. The invader knows that's happening, and they say, we're going to knock that out by injecting you full of with effectors. By the way, involved in that is type 3 secretion, which is a motif which is found in many, many bacterial pathogens. And as a result, the plants then have a second line of defense, which is the effector response to the second layer of response, the effector triggered immunity. Okay, so
can I just point out that part of this is also RNA interference. This is really important for virus infections, and viruses are sensed in ways that you suggested by pathogen-associated molecular patterns, and the RNA interference response is a big one for virus infections in plants, and that's been really well studied and some argue, was discovered. The whole RNA interference was discovered very early on in plants. Yeah,
that's right. It came from plants.
So I wonder if this is really innate immunity. I wonder if it was only invented once.
Well, plants and animals split off 1 billion years ago. Right. So there was something before that, and that something has been there for quite a while. So if you want to think it that way, you can say these systems existed before the split.
Yeah.
Interesting that the insects have only RNA-based immunity. They don't have protein-based immunity, whereas the plants apparently do, as you've said. So that must have been even earlier.
Yeah.
Oh boy, that's an interesting point. Yeah. I'd like to read a little bit more about that. That's interesting. Yeah. All right. So anyhow, with that for a background, we will then consider what this paper says. And this paper says that one of the MAMPs, that is one of the microbial, or not microbial, one of the pathogen factors, which is recognized by the plant, happens to be not made by the pathogen, but made by its symbiont. In the case of aphids, Buchnera. It's kind of neat when you think about it. You know, this is there's nothing known. There are no inducers by the aphids. There must be some PAMS-like compounds, but apparently either it's more complicated or hasn't been studied well, but we don't know of anything on the aphid which sets off the immunity. But a protein from the endosymbiont does exactly that.
But is that because they have to have the bacteria in order to be the aphid? You can't get rid of the bacteria in the aphid. It's an obligate mutualism. The bacterium is supplying something to the insect. The insect supplying something to the bacterium. So
it amounts to the same thing. It amounts to you can delegate the recognition stuff to the aphid, but it requires a little bit of gymnastics. Let me tell you why. The aphids, the bacteria in the aphids are not just floating around all over the place. They are contained in a cell called the bacteriocyte. Okay, that's an animal cell, a cell of the insect, which is, by the way, chock full of bacteria. There's just enough room for a nucleus and probably a few mitochondria. That's it. The rest is wall-to-wall bacteria. Now, bacteriocytes they sequester the bacteria. So how does this bacterial protein get through from the aphid to the plant? And the answer is, and I didn't know this is interesting, that in development, the bacteriocyte comes apart at one point. And let me see if I can find what this is all about, because it's an interesting story that I hadn't appreciated. And therefore, it is likely to move from the bacteriocyte throughout the hemocell, that is the
circulatory system.
Circulatory system, thank you, into the saliva. And this bacteriocyte turnover happens in a post-reproductive stage. Like you have to start out all over with making bacteriocytes. So this suggests that there is a mechanism for the bacterial protein to escape from the bacteria which are contained in the bacteriocyte and make its way into the saliva. And the saliva is, of course, what's injected by the aphid in feeding. And sure enough, if you collect enough saliva, you can ask this question. Now, how do you collect aphid saliva? All right. Well, first you got to get aphids, and that turns out to be relatively simple. I had to go back to several papers back because this is old hat. So they say as described before. And the as described before tells me that if you want to collect aphids, you take a plant which is infected with aphids and you shake it. You shake it over the surface and down come the aphids. And now you weigh them, and that tells you how many aphids you got. And in this paper, they're very proud of the fact that they use a lot of aphids. It turns out to be they use 100,000. But, if you have enough plants, you get 100,000 aphids. Yeah. You don't have to do much to get them. You don't have to pick them one by one.
Yeah, that would be tough.
That would be tough. So anyhow, this paper differs from previous papers because there are previous papers on this. Differs from previous papers in that they used 100,000 potato aphids. So, all right. So now you take these aphids and you make them feed in some kind of a little capsule, which is made of, I guess, parafilm or something like that. And you collect the saliva, all right? So parafilm pouches, as described before.
Well, that's how we get saliva from people. We have them chew on paraffin.
Yeah, but this is parafilm used as a
I know.
Yeah. Okay. But you're right. It comes in handy saliva wise. Okay. Now, you collect the saliva. What do you find in the saliva? Lots of bacteria, lots of proteins, And by the way, there are two kinds of saliva, but I won't get into that. Anyhow, you look at the saliva and you find out that some of the saliva comes from the aphids. How do you know that? Well, the aphid, you'd think that you have the sequence of the potato aphid, but you don't. So you do a transcriptome study of potato aphids, and that tells you more or less what kind of genes to expect. And from that, you can say that some of those proteins are of bacterial origin. Okay? And the big one among them, the main one, is GRO-EL. So let's go back to GRO-EL. GRO-EL it was discovered as a heat shock protein. Turns out to be a chaperonin, which is made in large amounts normally and is made in even larger amounts and comes into play when bacteria are shocked. GRO-EL is a highly conserved protein. It's very similar to again, heat shock proteins, which are found in the eukaryotic world. It's a fascinating protein. I think it's number 5 or 6 in abundance in E. coli, and number 1 in abundance in Buchnera, where it makes up for 10% of all the proteins. All right. So if you're going to pick a protein that's going to be recognized and serves as a recognition signal, you certainly want to pick on one that is abundant in such large numbers. Michael, I bet you know something more about GRO-EL that you want to say, do you?
I always have viewed it as the wonder protein. It's sort of like an usher, a molecular usher that escorts proteins so that they end up in the right spot. I mean, if you really sit down and think about the complexity of proteins and where they need to go, you can understand why in Buchneria there's an ever-pressing need for GRO-EL. And I think what's really neat that you're going to probably get to in this paper, so I won't steal your punchline, is how GRO-EL plays this role. And it's really about doing what the microbe needs done for the important or essential proteins so they end up in the right spot.
Yeah.
And bacteria can't live without it.
That's right.
It's essential, so it's important that it be broadly conserved.
Yeah. Now, however, from the point of view of the plant, this is just a chunk of chemical. And in fact, it's so much just a chunk of chemicals that you can boil GRO-EL. And since you can reproduce the phenomenon of setting off the defenses by using purified protein, you can do this with boiled GRO-EL. So here, GRO-EL is not a chaperonin. It's just simply a chemical that can be denatured in the denatured state, works just as well because some of the recognition material is present probably on its surface. So I gave away one important part of the paper, namely that you can show that GRO-EL is involved in setting off immunity by a couple of interesting experiments, and one of them is just that. You purify GRO-EL, you make it in E. coli or something. Yeah, you make it in E. coli, and you purify it, and you smear it on the plant, and sure enough, you set off immunity, and you can tell this in various ways, including lesser fecundity of the aphids. In other words, the aphids lose their sex drive or something like that. They don't reproduce as much.
But so that's one of the coolest things of this paper is how they assess the activity. They just look at the nymph versus adult ratio, and you ask, are they having sex? And the evidence of sex is babies.
Right. So fewer babies means that the defense mechanisms are operating. It's a good way, but they have other things too, but it's certainly a very neat one. The other way to tell that GRO-EL matters is you make transgenic plants with GRO-EL. And when you do that, you decrease the fecundity as well. So it works in a couple of ways. So this is about, give or take the paper. I didn't spend as much time on the paper as I did on the background, but the reason for it is, as you can tell, because I'm excited about learning about plant immunity. So to summarize, this is a story where the presence of an invader is carried out by determining the presence not of a invader element constituent, but one of its obligate endosymbionts. Unfortunately, there is no genetics on Buchnera, and so we can't make mutants of Buchnera to really nail it down. In a way, you'd like to do that with a mutant, and you can't do that because Buchnera, typical of endosymbionts, doesn't have much in the way of genetics. Would be very hard. It can't be grown, by the way. Obviously, it's a very reduced genome organism, and it can't be cultivated. And so, doing genetics with Buchnera is essentially impossible.
So you think that maybe making transgenic plants with GrowEL might be a strategy to prevent aphid destruction?
For potatoes and tomatoes and all of the other things that die in my garden because of aphids.
Of course, then they're genetically modified, and half the world will not eat them anymore, I guess, or half the U.S.
This is really a very big point, namely that plant breeding for disease resistance can easily include this kind of consideration.
Sure.
This belongs in that part of the conversation. So you learn a lot about practical things. This is important because it may lead to genetically modifying plants. That may be a dirty word in some circles, but I will use it anyhow. Plants can be genetically modified to become more resistant to invaders. And from a theoretical point of view, from a basic science point of view, it really illustrates evolutionary histories that are really worthwhile looking into.
I think it would be interesting to figure out what is recognizing GrowE in the plant, right?
Yeah. Sure.
And how does it get there? Because this it functions in the cytoplasm. They do point out that if the protein is denatured, it's even more easily recognized.
Right. Yeah. That's right.
Giving the impression that the bacterium has been degraded or something.
No. Well, maybe so, but I think it tells you that it's not the protein working as a chaperonin. It's not doing that. It's just working as a chemical.
One of the things that happened to me is I, like Elio, fell into the trap of plant immunity. So I went out and found a Nature review that occurred, I think it was written in 2006, and it's simply called The Plant Immune System, and it highlights many of the same things that Elio talked about. So I'll give that to Ray in the show notes if anyone's interested.
Well, then keep in mind that since 2006,
there's a lot's happened.
There's a lot more. So this is it's a big topic. It's really a big topic, and I plead guilty to having neglected it. So trying to make amends.
All right, thank you, Elio.
Sure.
The next second paper is from Michele.
Yes, and it's titled Immunosuppressive CD71 Positive Erythroid Cells Compromise Neonatal Host Defense Against Infection. This was published
we're still on immunity. We're still talking immunity.
That's right.
Yes. This was published in the journal Nature in December of 2013, and it is the work of Shakrola Elahi in the group of Sing Sing Wei at Cincinnati Children's Hospital and their team of another 10 scientists. So the question that Elahi and his colleagues tackled is, why are newborns more susceptible to infection? So you might think that there's a simple answer. Infants can't walk, they can't talk, they also can't fight infection. Their systems are just not mature. However, this group has discovered some really surprising and exciting new biology. They find that for a very brief period, newborns retain specialized cells that can actively suppress the immune response to microbes. Moreover, these cells, which we're going to find are an immature red blood cell, are really abundant in the GI tract. So as our TWiM listeners know, the GI tract has to become colonized by gazillion beneficial microbes. So the interesting interpretation that will lead to is that these specialized precursor of red blood cells are actually evolution's kind of uneasy solution to a classic good cop, bad cop story. We need to have newborns' guts be hospitable to microbes, but then people also and mice need to be able to resist infection. And so this mechanism is what we'll discuss in the paper.
Michele, I have a question before you go on.
Sure.
Did you get the sense of comparing this to something like fetal hemoglobin, where the newborn switches from fetal hemoglobin to hemoglobin once it is exposed to atmospheric concentrations of oxygen, and maybe Mother Nature has reinvented herself with this sort of thing?
I think that's an interesting parallel. Yeah, there's a big adjustment from the womb to life outdoors, so that it may be another example.
Huh.
So to investigate how why newborns are susceptible to infection, Elahi took advantage of Sing Wei's lab expertise with a mouse model of Listeria infection. So listeners may remember we talked about Listeria being an intracellular pathogen, and it's been used in the research labs for decades and is a great model for probing immune mechanisms. So he first needed to verify that newborn mice are also more susceptible to infection, just like humans. So in Figure 1, they infect either newborn mice or adult mice and with Listeria, and measure their survival, and sure enough, the newborn mice are more susceptible. And then they quantify the bacterial burden in the liver and spleen and see that there are like 4 logs more bacteria in the newborn mice. So confident now that they've got a good mouse model for studying this question, they then started to break down the biology and look in more detail. One experiment they did was found that if they took adult cells and transfused them into the newborn mice and then infected with Listeria, they weren't able to protect. So it didn't seem that the newborn mice were lacking something that the adult cells had. And for that set of experiments, and for the bulk of this paper, we're going to focus on a group of cells called splenocytes. So this is a general term that just refers to the gemisch of cells that are released when, in the lab, we macerate the spleen. So you just mash it up and release the cells. You'll get lots of macrophages, T cells, dendritic cells, red blood cells, and precursors of red blood cells, which are especially relevant for this story.
I have to say, that was a stunning result to me. I would have thought you put the adult cells in and that takes care of it.
Yeah.
It's amazing.
So it meant there's a story here. So then they got a really great clue from the next experiment where they used cells in culture rather than whole animals. So in Figure 1F, they took these adult splenocytes, exposed them to heat-killed Listeria, and showed that, as expected, these leukocytes can secrete lots of cytokine, in particular TNF-alpha. So this is a hormone of the immune system that tells other white blood cells what to do, and it's their readout for a robust immune response. So the surprising result was when they again took the adult splenocytes, the heat-killed Listeria, but they included splenocytes from a newborn. So by adding the newborn splenocytes, they now got very little TNF-alpha. The first clue that these newborn splenocytes were actually inhibiting the power of the adult splenocytes to generate an immune response. So they went on in Figure 2 to show that this suppression activity of these newborn splenocytes was dose-dependent, and they also showed that it could inhibit the maturation of T cells. So it seems to be a more general inhibitory or immunosuppressive activity. They also used some inhibitors of arginase enzymes and showed that you need an arginase activity, and that observation is going to be valuable for future studies of mechanism, but they didn't pursue it anymore in this study. So having demonstrated,
arginine.
Yeah, please.
Arginine sort of plays a special role here, doesn't it?
It does. They don't yet have a model, but did you want to speculate on that?
I don't know. I have no idea, but I've run into that. I've heard of arginine being important in immunity somehow.
Yeah. It's an interesting observation, and we can get to this at the end, but there's a widely used drug that is an arginase inhibitor, and that would be Viagra. So if this is important, we can imagine developing drugs that would be safe to use in humans. But let's get back to the story. So now they have evidence that if they macerate the spleen of newborns, there's a cell type or an activity that is able to suppress inflammation. So to figure out what cell it was, they used a common strategy in the immunology field, which is to separate cells according to the proteins on their surface. So to think about this sorting technique, imagine scanning a large crowd of people where it's very difficult to judge the talents of any one individual in that large crowd. But if instead we separate the crowd according to what they're wearing, we could have perhaps a group of people in big raincoats with boots and oversized hats, and we might think, firefighters. Or if we see others in big floppy white hats and white pants and white shirts, we might think they're cooks. Or if we see a group in bright plaid pants and polo shirts, we might think they're golfers. So that's the strategy that immunologists use. They can take advantage of antibodies that can bind to these uniform or surface proteins and pull them out of the population and then study those cells individually or study the complex population that's now missing that cell type. And that's what they did. So they removed cells that had the uniform protein called CD71. This happens to be a transferrin receptor. And then they tested whether the remaining population could still do this suppressive activity, and they found it could not. So they were then able to deduce that one of the cell types in that gamish from the spleen is a CD71 positive cell that has this suppressive activity. Now, there's very little known about this cell type. We know it's a precursor to red blood cells. There is one or two papers from a Russian group, like from 1970s, that described, that hinted at this. But basically, this is new biology for modern immunology. So to investigate whether humans have a similar cell type, they took advantage of blood cells isolated from umbilical cords that new mothers had generously donated to science. And they found the same pattern. That is, if they pulled these CD71 positive cells out of that complex population, they now found that the cells had lost the ability to suppress this immune response or inflammatory response. So encouraged by these results, they then went back to their mouse model and they depleted the CD71 cells from infant mice and then challenged them with Listeria and found that there was a correlation. So if you lose the CD71 cells, you actually become more resistant to infection. You have a lower burden of bacterial colony-forming units in the liver and spleen. So again, they were encouraged by this and wanted to test this idea further using a different approach. And here they took advantage of the observation that it was the infant newborn mice that were susceptible to Listeria, whereas the adult mice were not. So they simply analyzed mice at different stages of development, quantified how much CD71 positive C cells they had, and quantified their susceptibility to Listeria. And again, they find this nice correlation that if you lose the CD71 cells, you become more resistant to infection as you develop. So this was puzzling. We usually think that cells of our body provide a benefit, and yet here we find that if we remove these cells, the mice are more protected. So what could explain this puzzle? They thought about different tissues of the body and who's exposed to bacteria and who isn't. That motivated the next set of experiments where they isolated cells either from a newborn mouse intestine, which we know will be colonized heavily with microbes, and compared those to cells isolated from the spleen and from the lung, which by and large are sterile. And they found that only the intestinal cells were sensitive to this depletion of the CD71 cells. So the other cells would not then induce a large cytokine response. And importantly, they also found that their experimental system required that there be microbes in the gut in order to see this activity in these intestinal cells. So, for example, they could use antibiotic treatment to obtain mice that had little colonization in their gut. They could then not find this activity, or they could use animals that were raised in a germ-free environment. And again, they did not respond now when you depleted the CD71 cells. So, taken together, we're left with a model that that newborn mice have a transient persistence of this immature cell type, immature red blood cell, specifically in the gut, and it's sufficient to dampen the local inflammatory response, therefore making the gut more hospitable to colonization by microbes once the mouse emerges, or the infant mouse emerges from the birth canal and is colonized by microbes. So therefore, they're able to establish a symbiotic relationship with the microbial community, which we know is important for digestion, for generating vitamins, and also acting as a barrier to pathogens. But it is a trade-off. It means that during that window, when the newborns are benefiting from this immunosuppressive activity in their gut, it does mean that they're more susceptible to infection by pathogens.
But presumably, the newborn animal, whether it be a mouse or a person, is presumably breastfeeding, where they're getting passive immunity from the mother's milk.
Of course,
which
they also have acquired antibody from the mother passively, which protects them for 6 months or so.
Placenta, yeah. Right. But it's also the case that infants tragically are more susceptible to Listeria and E. coli, the two pathogens that they use here.
So let me ask you this question: Do you think that this magic CD71 and the associated effector molecule that's unique to this cell population will eventually be developed in order for us to begin to use it to, once we find the right microbiome mix, to give people the right type of microbiome that could make them recalcitrant to the effects of type 2 diabetes or weight gain or just generally proper nutrition, that the antibiotics that we're giving to our children today are thought to be depleting the good bugs from our system, and we don't yet have a real way of resetting us back to ground zero. So this CD71 may be a way, after you sterilize the gut, have the right microbiome magic elixir that you can give as a probiotic, and then reseed the system so that it takes by using a CD1 type derivative.
But the presence of CD71 makes you more susceptible right?
to infection. It allows you to get colonized, and then you can back it out as time goes on.
But remember what Laura Hooper said at the GM: throughout the adult life, you have to maintain the right distance, and the innate immune system is involved in that. Just to keep that 50 micron border between the cells of the intestine and the microbiome. So this is just the beginning. I'm sure it's not the only answer, right? It's going to be more complicated.
I had the pleasure of having a really great conversation with Elahi yesterday, and he is quite interested in whether this pathway accounts for why it's difficult to get a robust immune response to vaccines in infants. Perhaps if we could, in a controlled setting, reduce the CD71 activity in newborns or in infants, they would be more responsive to vaccines.
But I thought it only counts in the gut.
Yeah.
In the mouse model, that's what they have found, although they did see that activity in cord blood as well. It's not clear.
I bet it's more widespread because infants are susceptible to infections of many tissues, right?
Yeah,
not just viral infections can be disseminated in infants. Many years ago, we did an experiment with polio in mice. So if you take polio and if you take the vaccine strain of polio and inject it into a mouse, this is a transgenic receptor mouse, the mouse gets no disease. But if you take a neonatal mouse and put the same vaccine strain in, they succumb to paralysis in the CNS.
Wow.
So it's a pretty broad immunosuppression, if you will. And it may be multi-component. It may be this together with maybe missing other factors depending on the pathogen.
But it's exciting. This is a new cell type and a new activity that really has not been described.
It's very cool.
So I also was really interested in talking to Elahi yesterday that he made some observations in an earlier postdoc that really led to this. So he's Persian. He was raised in Iran, and where he got his bachelor's, master's in microbiology, and also medical training. And then he went on to do a PhD in microbial pathogenesis, in particular Candida albicans in a mouse model at the University of Newcastle in Australia. But then his first postdoc was he was challenged to develop a pig model for studying Bordetella pertussis. And when he arrived in the lab, they had been trying very hard to use 4 to 6 week old piglets to get them to colonize with Bordetella so that they could develop a better vaccine against pertussis, and they'd had no success. So one of the first experiments he did was to try younger piglets, so 3 to 4 day old piglets, and he found, in fact, that they were susceptible. So it was that observation, that memory that stuck in his head that the very young piglets were susceptible, but by 4 weeks they were not, that he retained. And then when he joined Sing Wei's lab in Cincinnati, had the opportunity to set up a new project. And so he went back to that idea and really pushed it with the mouse model. So it's a neat story. He, by the way, is as of January 1, is on the faculty at the University of Alberta and is setting up his own group now to pursue these studies.
Nice. I think it's really neat how there's a window of danger, right? You have to let in something good and to pay for that. But the assumption, I think, the evolutionary assumption is that you're going to be protected by maternal passive immunity, although it doesn't always work.
Right.
So arginine is an immunomodulatory compound, and you can actually go to the health food store and buy it as an immuno booster. I don't know if that does any good, but I know it well because it's a substrate of nitric oxide synthase, which is an interferon-induced enzyme, and that makes nitric oxide, which is a nonspecific destroyer of infected cells. And so that's the connection here in part, I think.
Yeah, it'll be great to see the mechanism once. Yeah. They've isolated this cell type, and a lot of potential here.
This is very cool. Yeah, this caught my eye. This is published in December, and caught my eye back then, and now we just got around to doing it. I think this is a really neat story with that, and he's going to have a lot of fun working on this in his own laboratory. That's great.
Yeah, and the idea that we could make vaccines more potent, I think, is really interesting. And there was a big story in today's New York Times about vaccination and that complicated public health issue.
Well, measles continues to rage across the country, and so folks are concerned.
Mm-hmm. There was a time when nobody paid much attention to science. At least that's how I view it. People got vaccinated and didn't complain about the kind of experiments that were done. And I guess it's good that the public's involved, but now it can be detrimental. We have anti-vaxxers, and now lately in the past year, people are really upset about the kinds of experiments being done with influenza, and it's unfortunate, I think.
Yeah, today's story also dealt with the economics. The cost of vaccination for a healthy adult or a healthy child rather has really increased, and so people. Practitioners as well as patients are having to make tough choices.
Well, it's funny. Last Friday night, I was surfing channels before I went to bed and ran across the comedian show by Bill Maher, and he was interviewing Marty Blaser, and poor Bill Maher was struggling with pronouncing the word microbiome. He couldn't pronounce microbiome. And he interviewed Marty Blaser, and the interviews are out on the net, so you can go take a look. But, Dr. Blaser did a tremendous service to the viewing public because he actually explained the microbiome in a very interesting way. Blaser was hawking his book, Missing Microbes, And he really did a tremendous piece for science literacy, because here's a comedian who's trying to make jokes, but at the same time, Dr. Blazer was able to advance the cause of science and actually teach a lot of people about the importance of the microbiome and antibiotic use, and how many antibiotics are being used each year in livestock feed. And it was really an interesting piece. So if you have time, and it was a free HBO weekend, so as I was surfing through the channels, I stumbled across Marty Blazer and Bill Maher. Who would have thought?
The other option you have is Dr. Blazer was recently on The Daily Show with Jon Stewart, being interviewed for the same book. So
yeah, he's out selling his book. So he did a tremendous job, and he's a great spokesperson.
So what's the name of his book?
Missing Microbes.
Have you guys, anyone read it?
Not yet.
Yeah.
It's on my summer reading list.
I heard him talk. He's terrific.
Yeah.
He's a great speaker.
We should get him on TWiM sometime.
Well, he's just down the street
from
you
absolutely.
At NYU.
Yeah, down the street from you at NYU, you bet. Well, thank you, Michele.
My pleasure.
I have a couple of emails I'd like to read. I have a follow-up from our last email, and this has to do with the paper or was it me? I discussed on plaques and bacterial biofilms and stress. This is from Dave, who writes, "Friends, please forgive brevity of pleasantry. Sleep is overdue. Fear presumptuous correction of trivia you already know, but RE stress and arterial plaques some points. Stress is not just perceptual emotive response to psychological irritants like commuting. The popular usage has been misleading us. It is bodily response to stressors, including many non-psychological events, such as physical exercise. Therefore, sudden blood pressure changes and even moderate exercise, at least when not begun with modulated warm-up, is a stressor to arteries and their plaques. Regular exercise benefits longevity in many documented ways, but burst of exercise can also rupture burst plaque deposits, as well as initiating arterial damage for initiation of the plaque site, releasing gruel into the circulation, and setting off big trouble as it clogs the smaller vessels. With bad luck, even well-modulated exercise might do it. Arterial plaques, Vinny, unless my ignorance has overtaken me, are distinctly macroscopic. You can definitely see them. Resident bacteria and biofilms are fascinating news to ignorant self, certainly microscopic, and must have distinct role in plaque-derived sudden circulatory injuries. The whole topic sounds thrillingly promising. So maybe Vince doesn't need a driver, and Michele had better watch out how she exercises, and we should all hope for fortunate expression of our genes and for magical cold iron transport or whatever. P.S. RE pre-proto or prokaryotes vis-a-vis eukaryotes. A while back, in a perverse moment, I skimmed an interview with a Bible literal believer who is a biology professor, department chair at Regency or Liberty University, arguing for a 6,000-year-old Earth and denying evolution dogma, which he illustrated by nonsense about pro-evolution bias being built into modern bio by sly usages of language such as pro and EU. Since pro means before, he said, the bias is a built-in acceptance that they came before true cells, thus adding years to the story of life and denying the biblical true age of life, the earth, and everything, including the evolution of species in any meaningful way. I betcha the pre- and proto-discussion mentioned by the letter writer has some place in this teapot tempestuousness. I'd guess protokaryotes protects against the suggestion that those cells may have temporal precedence, but are merely simpler versions of the fully formed thing or something. On which note, as long as PhD candidates aren't misled about jobs, the more educated, the better. Staff scientist is one good idea. So is forest ranger. So is RN. So is high school teacher or teaching college, etc. Besides, faculty need students. That's a good point. You can't have too many smart people, right?
No.
If they're teaching high school, that's great.
It's true.
I had some really smart high school teachers that got me into science, so I can only hope that there are smart people going to teach high school.
Michele, you were going to say.
I can say that pro and you has been maybe an important part of the argument. It hasn't.
I was going to say we don't spend a lot of time worrying that we have too many high school grads or too many college grads. So the idea that we have too many PhDs really rankles me.
As long as they realize they're not all going to be academic PIs.
Well, they don't necessarily want our jobs. I don't know if you've talked to them recently.
I have, but a lot are disappointed that they cannot. And a lot of them are saying now, okay, we don't want to be an academic PI. What can we do? And they say they're not getting the right training for other things. So I think that's where we have to put our energy,
Yep.
Another follow-up is from Tarwin, who writes, as always, thanks for the podcast. Weather jumping between 20 and 30 Celsius, depending if I'm in San Francisco or Palo Alto. Very quick idea question. Do you think there could be a link between hypothermia, anemia, and defense against bacteria in relation to it being a conserved trait in humans? What do you think, Michael? If you're anemic, are you better defense against bacterial infections?
Well,
It could be, but the question, it's a trade-off. You don't have enough energy to run and chase down your food.
Well, and maybe to run your immune system.
And run your immune system. I think it's good in practice, but hard to prove.
Yeah.
So it brings up a point that the author of the iron and stroke and heart attack paper that we did last week, David Davies made, and that is the observation that women have fewer heart attacks than men until they're postmenopausal, and then the numbers equal out. And he raised the interesting correlation that women, because of their monthly periods, are periodically anemic, and could that confer protection from the release of bacteria from plaques that may trigger heart attacks?
Yeah. And I seem to recall, I was reading a book about British royalty and how the wives of kings, they're in this constant race to produce a male heir. So they had lots of children, and there was a lot of bleeding. That was a way to treat things back then. And they typically became anemic rather quickly. But I'm not sure that helped them any. I think they're still quite susceptible to infection. This is all anecdotal, so I'm not aware of any study that's looked at that. And as you say, Michael, it would be really hard to tease out, right?
Yeah.
Rick writes, with respect to our episode on zombie plants, interesting that you focused more on the behavior of the zombie more than that of the vampire. My question is, what is the mechanism providing the awareness that you would assume would be necessary to comprehend the behavior of the creature being influenced or zombificated? Does the virus have a telescope to see what's going on at our human scale of existence? Another great show. Thank you. That was a paper where a virus was leading to zombie plants, right? They lost all their pigment. That was a while ago. So, no, the virus has no clue. The virus just does things that are evolutionarily selected to benefit the virus at that given time. So it doesn't really know what the outcome is at all. So the answer is no, not at all. Alice writes, an interesting study regarding the development of the microbiome of infants would be a fascinating discussion, I think, following your discussions of evidence for bacterial species in brain and urine. This is a story that broke a couple of weeks ago about the microbiome of the placenta. And she says might be a good pair with lung data. And this is another study on the microbiome of the lung.
Mm-hmm.
I took a look at the placenta article, and I think that we need to draw a distinction between, for me, microbiome implies like long-term colonization and a symbiosis, and then there's transient colonization.
Yep.
And I'm not sure they've established that there's long-term beneficial colonization in the placenta. Certainly transient makes sense.
Yeah. I heard some concern about that work when it first came out. What about the lung? Do you have any thoughts about that?
Yeah, there's I'm on the committee of a graduate student here who's with Gary Huffnagel, and they're finding microbes that you wouldn't expect in the lung. And I think it's, again, too early to know whether it's really a beneficial colonization or a transient, persistent colonization that's tolerated.
Yeah. The title of the lung paper is Lung Microbiota Promotes Tolerance to Allergens in Neonates via PDL1. PDL1 is Programmed Death Ligand number 1. And so that's a mechanistic. It's very much like the paper we looked at today with not just mechanistically, but multiple panel figures of flow cytometry and various assays, which is typical of nature. But there they have some data about the function, whereas in the placenta they did not. There was just a description of the placental microbiome. We all know that the gut microbiome can influence the development of immunity in the lungs. So, you don't have to have a microbiome in the place exactly. It can be somewhere else.
Right.
Yeah, we did that on the asthma paper.
That's right.
Very early on.
Yeah. Jeffrey writes, Doctors, I believe you missed the most important question in this episode. Can the N-formulated peptides that were found to induce pain in some bacterial infections be used in cooking? I smell a whole host of potential patents in the spicy foods marketplace. Thanks, as always, for the show.
But I think people eat the spicy foods because they get off on the pain because of the endorphins that are released.
Right. I don't know if these N-formulated peptides would taste.
I don't think they would taste. I think they would just suppress the pain.
Maybe.
Right.
Those N-formulated peptides were sensed by neurons, which then released chemicals that then were immunomodulators, right?
Right.
And so the control for those experiments was capsaicin, which bound to the same receptors on neurons. So, certainly if you eat capsaicin, you're going to get pain and
endorphins, which are pleasant.
Which is why they all love extremely spicy foods. And I think, if you eat enough of them, you either are killing the cells that are sensing the spice, and you're somehow titrating the brain's ability to sense the pain, but while at the same time dumping the endorphins.
Yep. Anyway, I don't think anyone's tried to. Well, the comment is tongue-in-cheek, of course, but
yeah,
it's an interesting thought. All right, one more from Shane. Hi, TWiM hosts. I have been a long-time listener to TWiM, TWIP, and TWiV, but I've never written to TWiM before. I really enjoyed episode 79, in particular, the story about using Listeria as an immunotherapy for cancer. I'm not very familiar with Listeria, and my knowledge of the immune system is limited to what I have heard on TWiV and Vincent's Coursera course. So this story prompted me for a clarification on a few points. Do you guys remember that story?
I do. Okay. That was my description of work from, Dan Portnoy described it, and it's a collaboration with Aduro Biotech.
Oh, that was from the GM, right?
Yep.
Yeah. Okay.
Yep.
1, if you can use the surface antigen from the tumor to stimulate an immune response against the tumors, why don't the tumors themselves already stimulate the required immune response? If the immune system isn't responding to the antigen on the tumor, why would it respond to the antigen secreted by Listeria? Does the antigen have to be in the cytosol to be detected?
Exactly. So you hit the point on the head. If the antigen gets into the cytosol, you trigger a very different type of immune response. It's one where cells can actually recognize and then kill infected or tumorigenic cells. So that's the difference. You have to have Listeria putting the antigen into the cytosol to trigger those cell-mediated immunity.
The answer comes from real estate. It's location, location, location.
Right.
2, if the Listeria has a mutated internalin, which won't allow it to attach and get into cells, how is it getting into the cytosol to stimulate the immune response? Or is it just secreting the antigen in the extracellular space, or is it only picked up and eaten by macrophages?
So if the bacteria have internalin, they can get into macrophages and also cells that are not phagocytic. So by eliminating that, the only cells that will take up the bacteria are macrophages. And then it's a different set of proteins that allow the bacteria to get out of the vacuole and into the cytosol. Those are pore-forming proteins. So that's the beauty of their design.
And 3,
it's the beauty of Listeria as a system.
Yeah.
Right, the genetics.
And number 3, if the Listeria is using these actin rockets to propel itself from cell to cell, how does it get out of the existing cell? Does the actin propel it through the membrane? Does it just break through both the current cell and the destination cell's membrane, or does it use the actin to get to the cell membrane and then use the internalin to get out of the cell and into the next one?
You've partly got it right. So they use the actin tail to push against the plasma membrane and push into the next cell, but they're still surrounded by double layers of membrane. They secrete then a different set of enzymes. They're called phospholipases, which allow the bacterium then to break down those 2 host membranes and then get into the cytosol. So they don't need the adhesin, the internalin, when they can make these rockets and push their way into the next cell.
Thanks for your amazing efforts on the best science podcasts on the internet. Keep up the great work. Yeah. Do you hear that, guys?
I think that's a great place to end.
It is. Best science podcast on the
Can't improve on
internet.
that, huh?
It's the end of TWiM 82, which you will find at microbeworld.org/TWiM and also on iTunes. And we love getting your questions and comments, as you can see. Send them to twim@twiv.tv. Michele Swanson is at the University of Michigan. Thanks for joining us today, Michele.
Thanks for having me.
And thank you for your erudite presentation of that paper. It was wonderful. Elio Schaechter is at Small Things Considered. Thank you for an education on plant immunity, Elio.
Oh, my pleasure, my pleasure.
And Michael Schmidt is at the Medical University of South Carolina. Thank you for the color commentary, Michael.
As always. And thank you for enlightening me about these wonderful subjects today.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for supporting TWiM, and in particular Chris Condayan and Ray Ortega for behind-the-scenes help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.